The overall goal of this procedure is to develop a simple technique for chromosome mapping of mosquito genomes. This is accomplished by first using imaginal discs of fourth instar mosquito larvae to obtain high-quality chromosome spreads. The second step of the procedure is to prepare fluorescently labeled DNA probes and unlabeled repetitive DNA fractions for blocking unspecific hybridization of the probe. The third step is to hybridize fluorescently labeled DNA probes to the chromosomes. The final step is to detect the chromosomal location of the probes using fluorescent microscopy.
The advantage of using imaginal discs over cell lines or brain ganglia is that the high number of banded spreadable chromosomes can be easily obtained from fourth instar mosquito larvae.
Using standard protocols, hatch mosquito eggs at 28 degrees Celsius. After 2 to 3 days, transfer second or third instar larvae to 16 degrees Celsius or 22 degrees Celsius incubators, depending on the species. The instar stage of development is based on head capsule size. When fourth instar become available, immobilize them on ice. Then, transfer the larvae to a slide with a drop of cold hypotonic solution. Under a stereo microscope, select larvae with oval-shaped imaginal discs for further dissection. The term imaginal discs is abbreviated as IDs.
Choosing the correct stage of mosquito larvae with oval-shaped imaginal discs is extremely